This is part 2 of chapter 4, and we were talking about isomers and just looking at this, the isomers here of C6H14. And we were talking about, I was mentioning that the different structures actually change the function and actual chemistry of these different forms of the same chemical formula. So 3-methylpentane makes a colorless um, liquid that smells like gasoline, and hexane actually is also colorless and smells like petroleum or gasoline. And you can see there's just really a slight change here, but there is a change, and that one change actually changes the boiling point from 64 to 98 degrees centigrade. So methylpentane has a boiling point of 64 degrees centigrade, and hexane's boiling point is 98 degrees centigrade. It ends up that hexane has it's very volatile. It's actually has industrial uses. It's used to extract edible oils from seeds and vegetables, and used as a solvent and a cleaning agent. Where methylpentane, I actually didn't find any use for it. It's actually quite toxic. Even touching it causes toxic reactions. So again, very simple change can change everything about a particular compound. So you need to be aware of the different types of isomers, and there are basically 3 types that we'll focus on. The first is a structural isomer. So the isomer that's structural have different covalent arrangements of their atoms. So they have different bonding patterns, and they look different. So you can see the difference there in these top 2, the pentane and the 2-methylbutane. On the other hand, we have cis-trans isomers, and with these, they have the same covalent bonds, but in terms of how the elements that are bonded are arranged, there's a difference. So when you have a cis isomer, you end up with 2 of the elements, these X's, on the same side of the double bonded carbons. And you'll find with cis-trans isomers, there's also always a double bonded carbon as a backbone somewhere in the molecular structure. And in this molecule, the trans isomer, so we have the same exact, it's to be a C2H2X2, whatever X happens to be. It can be just one thing like let's say an oxygen, or it could be something that's an entire grouping, like a hydroxide, an OH, or something even more complicated. It can be a total ring. But X just means that it's some sort of attachment, some sort of group that's attached. And for it to be a trans isomer, these 2 are identical and they're across from each other. And it's the same thing. These 2 are the same and they're next to each other. That means they're cis and trans. Okay, now with enantiomers, these are the ones that are called mirror isomers. So, and that makes it very simple in terms of the definition, because essentially if you put a mirror between them, it's as if they're looking at each other in the mirror. And so they are mirror images of each other, and you can see here it's the right and the left version. This would be the left isomer, this would be the D or right isomer. So that's important. In practice, taking a look at this one, I think this is probably the most difficult one to grasp and to make sure that you're getting the right configurations for this to be an enantiomer. So make sure you study some of these examples in the book and also maybe look up some online, because you do want to be able to recognize this one. I think this one gives the most trouble to people on tests. So keep in mind here how it has the exact same attachments around the carbon, but they are just mirrored from each other. Okay, so it says if you put a mirror down the middle. Okay, here's another quick animation that's going to discuss these isomers. Okay, again, these animations are short and sweet. One important thing about enantiomers is that they have a very big role to play in pharmaceuticals. Um, these mirror image isomers are actually really common, and most biomolecules or biologic or organic molecules actually end up with enantiomers of some sort. And sometimes, even when we eat something or when something enters the body in the form of a medicine, it can change from one enantiomer formation to the other. And that can actually cause problems or it can be beneficial. But some examples of where this comes into play in pharmaceuticals is, for example, with ibuprofen. So ibuprofen, which is like Advil, is produced industrially as a racemate. So two enantiomers of ibuprofen can occur with the potential for different biologic effects and metabolism for each of these enantiomers. So only one form is actually beneficial. And so what they had to do some testing to find out which one would be the best. And what they found is that different formulations of ibuprofen could actually affect how well it works. But what they found is there's actually an enzyme, isomerase, which isomer ACE, so it's an enzyme that works on isomers, actually can convert one form of ibuprofen to the other. So they could actually produce the inactive form, and the body would actually use its own enzymes to change it to the active or effective S version of the isomer. So these two different enantiomers in ibuprofen and many other cases can have very different effects on the body, and usually only one is biologically active. But it really shows how human organisms and all other organisms as well are very sensitive to even subtle variations in molecules. And so again, the variety of molecules that we get with carbon combining with different elements really is important to life because it is even the most subtle differences can make a huge difference in how it works in the body or how the body treats it. So this idea is going to come up a lot throughout this course that the form, which is the structure, affects the function. So structural differences create important functional meaning to life. So, for example, we have the amino acid alanine, and there are two different isomers of alanine. There's L-alanine, which is used in proteins, but there's also a D-alanine that's not used. And in terms of medicine, the L version can be active, but not the D version. So in this case, we have L-dopa and D-dopa, and we're actually going to look at a quick animation about this one as well. But one of the times when it says sometimes there are tragic results, sometimes unknowingly what has happened in the past is a drug is given in one form, but then the isomer of it actually is converted within the body, like we were discussing with ibuprofen. And that actually is what happened with thalidomide, which was actually prescribed to pregnant women in the 50s and 60s to reduce their morning sickness, and it was effective to do that. But its stereoisomer, which got converted in the body, actually caused incredibly sad birth defects. And there was a term called thalidomide babies to describe all these children that were born with these birth defects. And mostly they were missing limbs, particularly their arms. So something as simple as a stereoisomer, which again is the enantiomer, um, can cause huge differences in how the body actually metabolizes or allows a chemical or organic compound to work in the body. So this next video or animation will look at L-DOPA. So again, very simple. If you look at these two molecules, it looks like if you just flip them around, they'd actually be the same one. But they're actually stereoisomers or enantiomers, the same thing. And because of that difference, one is effective in helping prevent a very severe illness, and one is not. So it's pretty mind-boggling, actually, to see how specific our organisms are in terms of handling these organic molecules. So fortunately, carbon does allow for a diversity of molecules. Because we talked about hydrocarbons being the backbone of so many of these organic molecules, but carbon does bond nicely with all sorts of other molecules like oxygen or nitrogen or sulfur. And this allows there to become more and more properties that organic molecules can have. So it ends up that the properties of an organic molecule or biomolecule depend on the carbon skeleton and then on the chemical groups that are attached to it. So this would be one example of that, and it's actually an amazing example. But estrogen is also known as estradiol, and testosterone are both steroids that have a common carbon skeleton. And they have the form of 4 fused rings. And these sex hormones differ only in the chemical groups that are attached to the rings of the carbon skeleton. And so I'm going to show you a picture of just how different they are. But in terms of, before I do that, think of the difference that these two hormones make in humans and then actually most organisms. The difference between estrogen and testosterone and the balance of these two steroids within a body and within an organism determine so much about what happens in the life of that individual organism. So this is a picture showing the difference. So quickly, can you spot how many differences there are? Yeah, hopefully you can see they're highlighted in blue. There are only 2 differences. So the carbon here is double bonded to an oxygen, and this carbon here has a single bond to an oxygen or a hydroxide because it's an oxygen with a hydrogen, and then it has a double bond to its adjacent carbon. So it still ends up with 1, 2, 3, 4 bonds, but there's that substitution right there. And then here we have instead of there being a double bond here, there's another carbon group that's been bonded onto this one spot. So those 2 differences here and here and here and here are the difference between female and male. And of course, they are determined by the genetics as well.